Back to recording. And back to the slides. Let's go down. There we go. All right, back to it. Bacteria, archaea, and eukarya, 3 domains of life. We're going to focus on bacteria right now. So, very early on, it seems like the earliest type of bacteria are hyperthermophilic. So that means that they like heat and they like it to be really hot. So what that suggests is that the early Earth was very hot. And so these organisms adapted to that environment. And that also means that we can study their adaptations in the modern day hyperthermophiles to understand more about how Earth gave rise to life. All right, we're going to start with the Proteobacteria. This is the largest, most diverse group of bacteria that we are currently aware of. I feel like that goes as like a in parentheses for everything we're going to talk about today. As far as we know, this is the largest, most diverse group. These are all gram-negative. So as we go through today, I'm going to be talking about gram classifications and pay special attention to exceptions to rules. So you'll notice here we have alpha, beta, gamma, delta, and epsilon proteobacteria. So in your head, categorically, if you just want to give them a letter, that might make it easier for you to remember instead of alpha proteobacteria, if you just have A or something in your head, in your notes. And again, just keep in mind that we're using ribosomal RNA sequences, specifically the 16S, the small subunit, to draw these relationships. All right, so we're going to start with the alpha proteobacteria. These are phototropic purple bacteria. They're going to undergo anoxygenic photosynthesis using a pigment called bacteriochlorophyll. Anoxygenic photosynthesis means that they are not producing oxygen. Whereas organisms like the cyanobacteria will produce molecular oxygen from carbon dioxide. Not these guys, though. So as we talk about large groups of organisms, I think it's helpful to think about how these organisms play a role in different nutrient cycles. These are going to play a role in the nitrogen cycle, so they can have symbiotic relationships with plant roots. So organisms within the genus Rhizobium and Nitrobacter play a role in the nitrogen cycle rather. So those are symbiotic organisms, meaning that they work together to help each other out. We also have some pathogens. Pathogens are, by definition, going to hurt a host, so they're going to extract nutrients from a are transmitted by animals. So we're going to call these vector-borne diseases. So in this case, a louse or a tick is going to carry the pathogen. You get bit by it, so you have been given it from a vector. You didn't pick it up from the environment. Think about mosquitoes and malaria, West Nile virus. The mosquito is the vector here. So it's a moving transporter of a pathogen. And we also believe that this is the origin of eukaryotic mitochondria. I'll ask you again, what is the process evolutionarily that gave rise to mitochondria and chloroplasts. What was it? Endosymbiosis. So endo meaning within, and then symbiotic. So sym meaning together, and then bio, life. So together, they are able to give rise to a new form of life. Endosymbiosis. I think I've talked about that half the lectures. Expect to see something about endosymbiosis. All right, so there's the alpha proteobacteria going through the beta proteobacteria. So these are mostly aerobic. We're going to find these in wastewater and soil. Again, playing a role in the nitrogen cycle here. Some organisms within Thiobacillus, so that's a genus, can play a role in the sulfur cycle. If you want to know the hardcore chemistry of this, your book has these definitions. I just want you to know that we have bacteria that are involved in the cycling of nutrients. So maybe at the end of today, what I'll do is I'll make some sort of a slide for Wednesday where I categorize organisms according to which nutrient cycle they participate in. I think that would be useful. Bordetella is a genus that can cause pertussis. You heard another name for pertussis? The whooping cough, right. And then Neisseria is a genus that can cause meningoencephalitis. Big word that means an infection. So itis, meningi. These are the protective layers around the brain and spinal cord. And then enceph usually refers to the brain. So this is an infection of the protective layer around the brain. So as I'm going through, I just want to give you all indicators. So it's like, yeah, we're just going through and having facts about all these, but what should you draw from each of these slides? So of course, I just want to teach you all about microbiology, but then things that you should draw away as you think about what to expect on the exam. So these alpha proteobacteria have this pigment, that's special, and oxygenic photosynthesis, that's special. So that would be the one thing that I would take away from this slide. Right? Think like, what is a distinguishing characteristic that I should know for each of these groups? All right, for the gamma proteobacteria, these are going to go through anaerobic photosynthesis. They're going to do this using bacteriochlorophyll. And we're going to find them in oxygen-depleted water, so like ponds off in the distance or something. And these can store sulfur from hydrogen sulfide gas. I was reading about why they might store sulfur, and I read that that could be to produce sulfuric acid. Why would they produce sulfuric acid? It could have to do with keeping their internal pH regulated, or potentially to deal with other microbes in their environment. I would need to read into that. But they form what's called sulfur granules, if you're interested. Whenever you see enteric, think gut. So some of the gut pathogens, like Salmonella that causes food poisoning, Shigella, Escherichia coli, all of these fit within the gamma proteobacteria. Are any of these gram-positive? No. Right, because we said all the proteobacteria are gram-negative. So let's say that I just give you a series of the alpha, beta, gamma, and epsilon proteobacteria. Which of these is gram-negative? You would choose none of the above. or gram-positive.
I was like, wait.
That's what I think. Like, I need the eye contact. Right. Thank you. So let's just repeat that. If I were to ask you, which of the following is gram-positive, alpha, beta, gamma, or epsilon proteobacteria, you would choose none of the above because they're all gram-negative. Yersinia pestis, we've talked about a little bit. This is what caused the Black Death or the plague. And this is still present. I think there's a study that came out not too long ago. They found this on a New York subway. And then Vibrio is a genus as well as a morphology for bacteria cells. And this can cause cholera. 2 questions about Vibrio. How would you describe the shape of Vibrio as a morphology? What's it look like? It looks kind of like a kidney bean, right. It's kind of like a rounded kind of moon shape, kind of like a big, bold C, kind of like a bean. Okay, so great, good job. And then going back to chapter 1, so going back 2 weeks ago, do you all remember who it was that eradicated within, I think it was London, a cholera outbreak from a sewage-laden water pump?
John Snow.
There you go, John Snow. Somebody's been studying. That's awesome. Good job. Okay, and then Pseudomonas, this is also an organism that is pathogenic, and this is associated with cystic fibrosis. Pseudomonas can cause pneumonia in people. It forms biofilms in lungs. Okay, a little bit about the delta. So these include the aerobic Mycobacteria. Mycobacteria are special in that they form these multicellular organizations. So they're going to start with a biofilm, and then that biofilm is going to arrange into like a stalk and then some sort of a fruiting body shape. So this isn't a multicellular bacterium, but we get these kind of specialized cells within a cluster of Mycobacteria. So it's multicellular-like, so that they can produce spores. I'm not going to hold you accountable for the life cycle. I was going to show a figure. I think I might have one. Just know that Mycobacteria are special because they can form a multicellular-like fruiting body to produce spores that we're not seeing in other organisms. So these play a role in the sulfur cycle, and then these are going to be the bacteria that are responsible for rusting. One pathogen causes tularemia, or the rabbit fever, and then something you all might see in hospitals is Acinetobacter baumannii. People with immunocompromised, so immunocompromised patients, people with HIV, people on chemotherapeutic drugs, they are susceptible to this pathogen. And then the epsilon proteobacteria, Helicobacter is one genus, and that's going to cause stomach ulcers. It's also associated with stomach cancer. And then Campylobacter is going to cause enteritis, or like small intestine inflammation. So enteric refers to the gastrointestinal system. Enteritis is inflammation of the gastrointestinal system. Gastritis would be stomach inflammation. So yeah, like gastric ulcers, stomach ulcers. Enteritis is going to refer more to the small intestine, which is right after the stomach. I'm really looking forward to this body system stuff at the end of this, where we get to talk about anatomy too. This is going to be fun. We're going to get to use all this. We're learning it now so that we can talk about it later. I promise it's going to be worth it. All right, and then some of these epsilon proteobacteria are going to live in water, so maybe near hot areas, maybe near cold areas. So they have a diverse array of environments that they can live in. All right, so those are all the gram-negatives in the proteobacteria. Now we're going to talk about some gram-positive organisms. So we have two big groups of gram-positives, and the way that systematicists have organized them is according to how much GC content. So keep in mind, in a genome, we have A, T, T, and G. A is going to bind with T. G binds with C. There are 2 hydrogen bonds between adenine and thymine. Between guanine and cytosine, there are 3 hydrogen bonds. So that means that there's more strength between G and C than there is between A and T. Why would that be important? What could lead to a bond breaking? Think about what causes sterilization. Heat, right. So if you have more GC content in your genome, that means that your chromosome is more resistant to heat. So higher GC, if your DNA is stuck together better, then you are going to be able to survive in a more broad array of environments, specifically heat. Heat and pH. Big time heat though. So the actinobacteria are generally going to have a larger GC content. So you find these in the soil. Some of them are going to produce spores. Do any gram-negative bacteria produce spores? No, they don't. So just, if we're just riffing on these potential exam questions, which of the following groupings produce endospores? Alpha, beta, gamma, epsilon proteobacteria? None of them, because they're all gram-negative.
I thought you said the delta proteobacteria have like the fruiting body to produce spores.
Oh my gosh, you're paying attention so well. Okay. Very good. Tell you what, give me 1 sec. Let's pull out this textbook. Okay, so the textbook does not say anything about delta proteobacteria producing spores. I found that in an online resource. So let me double check on that. Really, really good question. Thank you. Who brought that up?
Me, Sabrina.
Okay, great. Thank you, Sabrina. I appreciate you very much. So I'm going to look into this, and then I'll email the class about that afterwards. So the question was, do the delta proteobacteria produce this fruiting body to produce spores? If they do, then they would not fit that gram-negatives don't produce spores. So I'm going to look into that. Very good question. All right, so within the gram-positives that can produce spores, the actinobacteria fit within this organization. They're playing a role in the carbon cycle. And Streptomyces is one genus found here. What is special about Streptomyces? What do we get from them? Keep in mind these are soil bacteria. We talked about it last time. What did we talk about last time? Thursday. Yeah, antibiotics. A lot of antibiotics are derived from Streptomyces.
again?
Yep. So Streptomyces is a genus of actinobacteria, and we derive a lot of our modern day antibiotics from Streptomyces. So then on Wednesday, maybe we can go through and talk about, as we kind of build these bridges between chapters, which of them they do produce. Okay, Mycobacteria fit phylogenetically within the actinobacteria, but they are not gram-positive. I will argue to the end that these are not gram-positive organisms, and let me explain why. You can't do the gram stain protocol on Mycobacteria. It doesn't work because of their special waxy layer on the outside of these cells. So if the gram stain doesn't work, you can't classify an organism according to the stain, because it doesn't work. So you can say that they are related to gram-positive organisms, but they are not gram-positive. They are acid-fast positive. Acid-fast is a test that says whether or not you are a mycobacteria. That is what acid-fast test does. It's going to look for that waxy layer called mycolic acid, mycobacteria, mycolic acid. That is what acid-fast does. It's going to say positive or negative for mycobacterial species. That is the only reason that we're going to do an acid-fast test. Have you all looked into your lab syllabus to see if you do that?
Yeah, we do that.
Very good.
The gram?
Gram, endospore.
We do gram stain.
Okay, that's great. So that's going to make going into the exam better. It's going to become more real when you see it, I promise. Okay, so that was one group of the gram-positive organisms. These have high GC content. Those that tend to have lower GC content are known as the firmicutes. One genus that does produce spores is called Bacillus. And this is what the Endospore Test is going to look like. So within the Endospore Test, you have two stains. You have a red and a green stain. The green stain will stain your endospores. Vegetative cells are going to be red. So you can see everything that's green inside here is a spore, and they're going to be within a red vegetative cell. These are obligate aerobes. So where would these be in thioglycolate medium? At the top. Would they be at the bottom at all? Nope, they'd die. All right, so this is Bacillus anthracis. This is going to cause anthrax. And then Bacillus can produce antibiotics such as bacitracin and polymyxin. We talked about these on Thursday. Which type of antibiotic are these? What? No. You tried though, that's all I want. These are peptide antibiotics. And keep in mind that these were mixed in with some other antibiotics, and these are commonly used in ointments like neosporin and polysporin. Clostridium is another genus. This is an obligate anaerobe. It can cause tetanus, botulism, gas gangrene, and then Clostridium difficile is associated with gastrointestinal complications. Botulism is a foodborne illness. And then tetanus, we acquire from getting like rusty bits in our body. Okay, Staphylococcus. Let's keep in mind that this is both a genus as well as a cellular arrangement and morphology. Okay? As the arrangement and morphology, how would you describe Staphylococcus? So what shape are the cells? They're round. And what is their arrangement? Yep, these are in a cluster. These are going to be the ones that look like grapes. Okay, so Staphylococcus, again, this is a genus as well as a arrangement and morphology. If it's italicized, we're talking about the genus. So, methicillin-resistant Staphylococcus aureus. This is an antibiotic-resistant strain that is going to spread through hospitals, through communities, and this is why we need to be very prudent with our use of antibiotics. And from what I was reading, I think MRSA originated in someone not receiving antibiotics, just through mutations alone, and then spread like crazy. Streptococcus is a genus as well as also a morphology and arrangement. These are going to be chains of round cells. Strepto chain. And so we can split these into two different groups, and I've included this image here. So if we take a plate with media, sometimes we'll put blood in it, and so that's why it has this red color. Hemolysis is rupturing of red blood cells. So if we put these two groups, alpha or beta hemolytic strep, on a plate with blood, we can either see kind of like a greenish color. So the green color is going to come from partial hemolysis, so rupturing some of the red blood cells. It's going to, I guess, free up the heme that's inside and kind of turn it like a green color, as opposed to complete hemolysis, where we see this clearing outside of the B, the beta. So what they did is they took their loop and then drew a beta with bacteria. I think that's kind of cool, right? So this shows that they are very good at rupturing red blood cells. Why would they do this? Inside of red blood cells, we have hemoglobin, right? Hemoglobin carries oxygen and CO2. It contains iron, and a lot of bacteria require iron, so they will rupture our red blood cells to take our iron. That's why people with chronic infections can be anemic. So anemia is low iron in your blood. That can be a symptom of a chronic infection. Mycoplasma, these are going to lack peptidoglycan. And the book mentions how these have such a small genome that people think this is probably the smallest that a genome can get in order to maintain life. So I think that's interesting. I do want you to know that. These are very, very small organisms. So these can cause pneumonia, maybe some urogenital infections. And where have we heard of lactobacillus for foods? Yeah, dairy products. So lactose is a milk sugar. So these you're going to find as your like live cultures in your Greek yogurts. And then I don't know how old everybody is, but if you drink beer and you like sour beer, it's a very acidic beer that's produced using lactobacillus as well. So it's going to eat sugars and make the beer just crazy acidic. And Enterococcus, another genus, and this is going to cause gastroenteritis, so stomach and small intestine inflammation. time. Okay. Cyanobacteria are photoautotrophs. Somebody tell me what a photoautotroph does. To do what? To make organic carbon sources from CO2. So we're going to make glucose from carbon dioxide using the energy from light. And we're going to produce molecular oxygen, O2, as a byproduct. So when we say that they're the primary producers of organic molecules, they're taking gaseous carbon and turning it into glucose. The book mentions these stromatolites. So if we have some cyanobacteria in a biofilm, they can capture some debris. So let's say they're forming their biofilm and they land on a little pebble. Well, they can end up calcifying that pebble just through letting out some of their byproducts. It calcifies and then becomes trapped. And then I guess that we've used the dating techniques for those stromatolites to figure out when these organisms were around, so how far back they go. That's why I think they talked about stromatolites, because it seems relatively unimportant unless we're talking about evolutionary origins. So we think that cyanobacteria are what allow us to be able to use oxygen. So they took that early high CO2 environment and turned it into a higher oxygen environment with less CO2. And these are gram-negative. All right, y'all, we have just a couple more slides. Hang with me here. So another group of organisms is the Chlamydia. We've heard about Chlamydia before. So these are special because they are obligate intracellular parasites. You're not going to pick up Chlamydia on a desk. You're going to get it from someone else. So it has a limited metabolic ability, which is why it requires a host, a human, or some sort of other animal to survive within it, because it can't do everything on its own. For example, it is non-motile, so what does it not possess? It does not have flagella. Flagella are how bacteria move. And it is gram-negative, which means it doesn't have LPS. So I say it's gram-negative, and then we classify gram-negative as having LPS. Chlamydia are weird in that they don't have peptidoglycan. So we can call it gram-negative, but it's not like a classical gram-negative where we have LPS on top of peptidoglycan. We have plasma membrane LPS, which is like a lipid fatty layer, right? So the book says that these, like during their binary fission, there's a little bit of peptidoglycan in the septum of dividing cells, but you're not going to find this in their cell wall. So these can cause pneumonia. So the species would be Chlamydia pneumoniae. That's also implicated in heart disease as well. And then Chlamydia trachomatis is going to cause a sexually transmitted infection, chlamydia. Spirochetes are these round, spiral, kind of spring-shaped bacteria shown in this figure in the bottom right here. So you're going to find these in muds, in insect digestive tracts, and then in our mouths. So going back to, again, chapter 1, whenever Leeuwenhoek was looking at those animalcules, if you look at the figure, you'll see a spring-shaped organism. You probably saw some of these spirochetes because he looked at dental scrapings, and we do have spirochetes in the mouth. They're pathogens. They're going to cause gum disease. And these are special because they have a special type of flagella called an endoflagella. Can you all remember where an endoflagellum is within the cell envelope? What? Yep. So it's inside, what's the space called? Periplasm. Yeah. Within the periplasm. So that's going to be between, I believe it's going to be between the plasma membrane and the cell wall. But either way, it is within the cell envelope. It is not an external appendage that's going to spin. So this is going to allow them to have this corkscrew fashion. Remember, this is what causes the bullseye in Lyme disease, because they're really fast, and they're even faster than our immune cells. So they can outrun our body, which is pretty cool. Scary, but still cool. And so we said these are found in the mouth. They can play a role in causing gum disease, periodontitis. They can also cause syphilis. 10 gold stars for whoever can tell me what causes syphilis. Treponema pallidum. You don't need to know that. Yeah, right. Avoid it. And then also Lyme disease, that organism is Borrelia burgdorferi. Don't need to know it. Just know that we find spirochetes in different places. In sediments, in the mouth, in urogenital tracts, in vectors like ticks. All right, and then the last group of bacteria that we're going to talk about are the Bacteroides. And your book talks about the relationship of these with obesity. So what they mentioned is that they did studies with mice and humans. So they looked at the gut microbiome. So which bacteria are in the guts, the intestine, of humans and mice that are healthy weight and obese. They found in both species, humans and mice, that obese animals had a higher percentage of firmicutes relative to the Bacteroides. And they found the opposite was true in healthy weight people. People who are not obese tend to have more Bacteroides than firmicutes. So we don't know if this is a cause, if it's just associated or what, but noticing something is the first, it's a jumping off point to asking questions and then following through with the scientific method. You've got to notice it first, make an observation. All right, 2 more slides and then we are done. As far as what you need to know about Archaea, I want you to know that there are 2 major classes of them, or categories rather. The Crenarchaeota and the Euryarchaeota is going to be on the next slide. Within the Crenarchaeota, know that these like really hot, acidic environments. So things like hot springs, these are going to tend to also be acidic. I want you to know that these are not bacteria. These are their own domain. And then within the Euryarchaeota, this is our last slide for the day. Some of these are Methanogens. So these are going to be in animal ruins, so something like a cow. We can find these in some mammalian intestine, insect hindguts. So you'll notice that these are going to be low oxygen environments. Our halophiles are going to require oxygen, and so halophile means they really like salt. So salt's going to be really abundant in the external environment. What's that going to do to the inside of a cell? Keep in mind that it's mostly water. Yep, so by osmosis, the water is going to go from inside the cell to outside the cell to deal with the high solute concentration outside. So what these organisms do is they will accumulate salts in the form of potassium chloride inside their cells, so that way it keeps the water inside their cytoplasm from moving out of the cell. And then these can also generate some ATP from light using retinal and bacterial rhodopsin, which is a photosynthetic pigment. And retinal is a protein found in our eyes. And then this last group that we're going to talk about, the thermoacidophiles. So these have special components of their cell membrane that help them deal with hot environments. Keep in mind that with heat comes movement, right? So a membrane, keep in mind, it's this mosaic fluid model where things are always moving. It's not rigid, it's not stuck in place. So things are going to move faster in a membrane with high heat, so they can regulate that by stabilizing it with these lipoglycans and lipoproteins. You don't need to know the names of anything, just know that they do certain things with their cell membrane to deal with the heat and the acidity. All right, y'all. So that is it for today. And so like I said, tomorrow we are not going to do chapter 12. We're going to do chapter 13. So we're talking about viruses tomorrow. We're going to skip the eukaryotic stuff. If I really want to come to it, I'll think about it over break. So between now and Wednesday, email me with all the questions you have. We'll do an hour-and-a-half review on Wednesday. It'll be really good. Let's study hard, study well, and learn a lot. Online people, do you all have any questions?
Nope, not right now.
All right. So I'm going to go ahead and end this recording. If you all need anything, feel free to reach out. I'm available through HC meetings. We can do one-on-ones throughout the week. Just reach out, let me know what you need, and I'm here for you, okay? Thank you all for your time. Yeah.
I had a question.
Okay.
So for the essay, when we're typing it up, how many, like, do you want it double spaced so you can read it easier?
You know, Leanna, is it Leanna or Liana?
It's Leanna.
I'm sorry, can you repeat that again? Leanna?
It's Leanna.
Leanna. So you can do it double spaced or single spaced. I'll leave that up to you. It makes no difference to me.
Okay, thank you.
Yeah, you're welcome. And then Sabrina, again, really good question earlier. I'm going to look into that as soon as I get home today, and I'll come back tomorrow with an update for you. So thank you for that.
Thanks.